Okay, recording is on. All right, so yeah, the other day or yesterday or oh my goodness, what Monday? Here we go. We were talking about energy and cells. I kind of want you to think of this big picture and little picture. I know. Actually, that's very helpful if you can do that. You know, if you're looking at the trees and you miss the forest, you kind of miss the overall idea. So remember what's going on here is cells are organized systems, and all organized systems, whether they're living or not, like the solar system and the universe, all that, they need energy to stay organized, because the natural state of things is disorganized. The natural state of things is completely separated, if you will. And if you're trying to get a visual, think about a piece of iron, an old piece of iron, maybe farm machinery that's sitting out in the field and getting rusty throughout the years and getting rusty. And after a while, I mean, literally with what, a few hundred years or so, it wouldn't even be there, would it? I mean, you could probably dig down the dirt and find evidence of iron and make some archaeological thing. But the point is, things do fall apart. So living organisms have to do that. No, gravitational attraction doesn't. It's a warp. Gravity is a result of a warp in the space-time continuum. Yes. If you imagine that space and time is a fabric, okay, and all of the planets and the planetary objects, stars and neutron stars, and all of them things have mass. If you held up a blanket, if you will, and you put a ball on it, it might make a dent kind of right there, right? And then maybe if it was a big enough blanket, you might put a bowling ball at maybe another corner of that blanket, and it would make a bigger dent. You might eventually put things close enough together that one of them would want to go to the other one. Now, the difference is that thing is also moving, so it's not going to go directly, it's going to spin. You know, we're spinning into the sun. Spinning, all of us, even the Pluto and the asteroids and all of the guys out there. So gravity is not an energy; it's a result of mass, right? You feel the gravitational attraction of the Earth because the Earth is pulling us in. Yeah. So did I answer your question? Organized by gravity. Trying to think if I've. Yeah, hold on. This system is organized by gravity. Yeah, let's talk about that in class, because that's an interesting question, because there's some things in there. Well, then you answer it. What's your answer? Or what is your thought on that? Because I'm curious. You got time in class. We can talk about this for a second. Ryan, are you in session? Yeah, you're in session, aren't you? You're on. I'm curious about the word you used, organized by gravity. I find that interesting, all right? No, that's interesting. You know what? That's actually maybe better. It gives me a chance to think about that. That's a very interesting way to say it. I didn't know if you said it by accident or you said it with some intent, but that's interesting. All right, so. All right, back to energy. What living things do to survive is, of course, they bring in a source of energy, food. We can't, some of us can make it, right? And we release the energy in that food, and we specifically release the energy of that food to make a specific molecule. We make the energy currency molecule, right? We make ATP. Oh, now it doesn't work. We make ATP, right? And it's real simple to think about how this works, right? Because the way you make adenosine triphosphate is you take adenosine diphosphate and you put a phosphate on it. Remember on the slide, there's a little I there. It means it's an inorganic phosphate. Don't let that get you off track there. But in order to make a smaller molecule, two smaller molecules in this case, into a bigger molecule, you're going to need energy. That's just the law of thermodynamics, isn't it? That's an endergonic reaction. So energy is, if that cost you must submit in order to get organization. Ooh, nope, I don't want to get off track. Anyway, so what living systems do if they want to survive, if they want to be efficient. And again, this is kind of a bio two things, but living organisms survive through time because they're very efficient, is they become efficient at using energy. They become efficient at turning reactants into products. And what I mean by efficient is you always have to put a little bit of a spark in. You're done. Stop making your bed. Thank you. You always have to put a little bit of a spark in, which we call the energy of activation. I mentioned in class, I think I used throwing a match into a gallon of gas, right? There's energy in a gallon of gas. That would be the reactants. The products would be, whatever it is from the gallon of gas, things that we don't really want to be emitting into the atmosphere, obviously. But nonetheless, you got to have a spark plug, right? Gallon gas isn't going to burn in your car without a spark plug. It's not going to burn in your garage unless you throw the match in. So you got to prime the pump a little bit. And so you see in the picture here what living systems do with these things called enzymes, which is what we're going to talk about the rest of the chapter. They allow these reactions to occur. This is the same reaction, but a lot less energy of activation. So they make living systems much more efficient, right? And they make living systems more efficient at doing this, right? Getting energy from food, catabolism. A friend of mine told me after class the other day, we were having lunch. He's the lab tech there, Scott. He said he always remembered it because cats tear things up. So I'm just trying to give you, ideas to help you remember catabolism from anabolism. So cats tear things up. So in catabolism, things are being ripped apart. So that bagel's being ripped apart, it's releasing energy, right? And that energy is used to drive that arrow that's moving up to the right, okay? And then, of course, that energy is not done. ATP, we'll talk a little more later, but ATP is, we'll talk a lot more about it, is volatile. Milk, milk if you left it on the counter, milk even if you left it in the fridge. Think about that as being my idea of what volatile means. It doesn't last very long. It's kind of a spastic molecule, a very sort of an unstable molecule. See, Mother Nature wants things to be stable. Mother Nature. We living things of Mother Nature take advantage of that. We can destabilize her by making ATP. Oh, ATP. It doesn't last very long. You only have a couple minutes in your body. But when it does break down, whoop, it gives off that yellow arrow on the right. It gives off energy for anabolism. So it gives off energy. And if you think about these things as being, wheels in a clock, they go round and round. Not a poem, they go round and round. So basically, the energy that's released from anabolism here could climb stairs, make me throw the pen around like I'm doing, or right, it could be recycled back here, couldn't it? Not to make ATP, sorry, but into another catabolic reaction. The energy that's released could do what we see down below. So the energy that's released could take, we could call these amino acids, couldn't we? And if we link them together through a peptide bond, but remember I said when we looked at ATP the other day, the chemical bonds are more than just physical. They're more than just physical. They're energy, right? They're energy. They involve energy to be put together. So what you're doing is when you digest your food, you release that energy. The only thing is, intermediate step, you do that. And that is that. And this, is an enzyme, a protein, a carrier protein, cell identity marker protein, any of those things that need to be made. Any of those things. Now, if you're sitting here wondering, is that how carbohydrates are made? No. Carbohydrates are made a totally different way. That's going to be the subject of 2 chapters from now when we talk about photosynthesis, okay? You can take carbohydrates and modify them, make them other ways. I'm thinking to myself, wait a minute, a little bit of AAP in there in case you have some of that. I don't want to confuse you. But all right, so there's this big energy cycle going around. So if you can make that whole process efficient, then you can survive better, right? This is the energy currency molecule again. This is what I talked about. This is ATP. It is a nucleic acid, technically, because of what it's made up of, but it doesn't have anything to do with genetics. It's energy currency. It's how your body spends energy. It's how your body, it's the common language of energy your body uses. You bring in different types of food, different energy contents, and so And they're all of them, whatever you eat, is used to make ATP. And then you use ATP for making proteins or climbing stairs or, whatever you need energy for, right? That's what you mean by currency. Currency, like, you got $5 in your pocket, you can buy anything that can buy $5 or that costs $5. You see those bonds up there, which they've labeled alpha. That's that little weird thing on the right. Then beta and gamma. Those bonds are not just something physical. They're obviously covalent. They represent energy, a lot of energy. It's hard to get a feel for the amount. 7.3 kilocalories. That's kilocalories, by the way. The stuff we eat in food, the amount is actually kilocalories. So the Big Mac is what, 700 or so calories, 700,000 calories, 700 kilocalories. And that's per mole, remember? That's all that has to do with atomic weight. Now, the minus, you might want to pay attention to that just because it makes it easier to understand when we see some other equations like that. The minus means energy is released. So it's an exergonic reaction. So the idea with the red arrows is when you break off that last phosphate, turn it into ADP like down below, what you do, as you can see, is you release energy, right? So you're engaging in a catabolic reaction. So you made ATP with a catabolic reaction, right? You took the energy from food and you made ATP, right? You were catabolic, releasing the energy from food, anabolic, making ATP, ATP being turned into ADP. That's going to be an anabolic reaction. We're going off, whoops, my hand's in the screen. We're going off this way, and now the ADP that was down below has undergone a catabolic reaction. It was made through anabolic, tore apart through catabolic. The energy was shuffled from the bagel through the energy currency to building a protein, climbing stairs, et cetera, et cetera, et cetera. Right? Make sense? Any questions on any of that? Right? It kind of puts all this together as I was talking about in the forest for the trees. Good. All right, Ryan, you're good on that? So let's talk about those things then that would lower that energy of activation, that would make you more efficient. And again, more details if you're really interested in the efficiency and organisms and all that in Bio 2. But nonetheless, this big blue thing is an enzyme. It's a bunch of amino acids, and it's folded because it has the right amino acids in the right place. So I'm hopefully repeating myself, right? And enzymes are specific, and they're specific because of shape, and they're specific because the active site only fits the substrate. Something that you call an enzyme's substrate is what binds to the active site and then is turned into product. Things that don't bind, things that aren't turned into product, aren't substrates. So you can't say, well, that's not the enzyme. Well, you could say that. But, okay, they're specific, right? Lock and key arrangement. You understand that. I don't need to give you an analogy of that. The induced fit model, though, is kind of cool. I always love the things in Mother Nature that means that it is random, because sometimes things aren't perfect, okay? So the induced fit model says, since you made this big, expensive enzyme molecule with a particular active site, maybe you didn't shape it quite right, okay? You got one amino acid wrong, we sort of twisted the top of it. Oh man, just a little bit, but it's close enough. And so the induced fit model allows for Mother Nature's fudge factor. Make sense? You see a lot of that when we go through biology, not just here. You see a lot where you go, oh, that's interesting. That system is designed. We're going to see it in genetics, actually. That system is designed knowing that it's a random thing, and sometimes bad things happen. Now, sometimes enzymes are so denatured or so misshapen, like when they're made, there's nothing you can do about it. That's just, well. But sometimes it's close enough, okay? But what an enzyme does, it's organic, so you should know what that means. But what an enzyme does specifically, it does this thing called catalysis, right? It undergoes catalysis. Let me show you what that is here just a second. I'll remind you, here's a good enzyme, the very complex shape. The name of an enzyme, can't remember if I went over this the other day, but if you see the name of this one, for example, pyruvate dehydrogenase. Remember in class we talked about things that end in OSE? that they're oftentimes they're carbohydrates. Here, things that end in ASE often are enzymes. So little cue for you there. And sometimes in the name of the enzyme that ends in ASE, it'll tell you what its substrate is. And sometimes it'll even tell you what it does to it. Like pyruvate dehydrogenase has a lot of info. You don't know what pyruvate is, but you will. We'll talk about it. It's a molecule, okay? And what pyruvate dehydrogenase does, it's an enzyme that takes hydrogens off pyruvate. Okay? Cool, right? Neat design. But remember, it's all about shape with proteins, and they're going to have the right shape in order to work right. Cool picture, too, of them in an electron microscope, too. What's this thing about specificity? Well, it's the active site, right? So the active site, the two substrates, the green one and the red one. So things that would not bind, or in just a second, we're going to talk about things that do bind. But things that do not bind, or things that bind but don't make products, are not substrates. By definition, it's the equation down the lower left. E plus S, an enzyme plus a substrate, bind. They make a complex, like you see in picture number 2. They're bound together. And then something happens, some chemical reaction. Enzymes can take two things and make one, can split one thing in half. They can take many things and put them together. They can change them. So in this case, it switched their shapes or something and made them different colors, right? It changed them. Those are the products. So the ES complex releases the enzyme plus the product. And then you notice it's a cycle. That's the catalysis part, or being a catalyst. A catalyst in science or in chemistry is typically something that causes a reaction, right, but does not get changed itself. So that's why it's important in this equation down below. It looks like a really simple equation, but this is the general equation for any enzymatic reaction, and there's as many enzymes as there are substrates, and there's a boatload of substrates, okay? But in any of those reactions, no matter the substrate, the enzyme always goes in and comes out the same way. It's the S, the substrate. Oops, now you don't work. How weird. Anyway, the S gets turned into product. Okay? The important thing that you understand about this, of course, is the reason that you could explain to me why. Why do you think it's important? Why do you all think it's important that an enzyme be of that type of molecule, like a catalytic molecule, one that causes a reaction but doesn't itself get changed? Now, what I mean by changed is it doesn't mess up its ability to do its job, which means it doesn't change its active site. It doesn't change something about its shape. Why do you think that would be a good thing? Well, I figure out, well, my pen doesn't work. I think I've used the word in class before. It's sort of an odd word, but I think I told you proteins are very expensive, didn't I? Didn't I say sometime in class that proteins are very expensive? And what I mean by that is they're big molecules with lots of energy. Yeah, I did. So does that help? If I tell you that, if I remind you that proteins are very expensive, and hopefully I don't have to remind you that enzymes are proteins. Okay, so why would it be important for Mother Nature, living organisms, to design this thing in such a way that it does have many uses, multiple uses? Think about your car for a second, right? Think about your car. Would you buy a car that lasts once? Yeah, there you go, right? You wouldn't buy a car that lasts once. You want a car that lasts multiple times. Now, much like your car, does the enzyme last forever? Forever? No. Why? Yep. You got it. Everything falls apart, man. Everything. Even enzymes. Now, they're expensive, so you're going to use them a bunch. I don't know. There's probably enzyme biologists, molecular biologists, that have numbers for how long an enzyme typically lasts. But yes, they have to be replaced. So, you start putting, and that's good, you're starting to put those concepts together, right? So we talked about protein shapes and how they're made and how specific. And this class is actually going to end in genetics, which has to do with building proteins, Remember, carbs and lipids, nucleic acids, those are made other ways. We're talking about building proteins here. That's the important thing, because the proteins are you. They're not only your enzymes inside of you, but they're you, all of you. They're what you look like, the way you work, whether you work well or whether you don't work well, okay? All right, now, one thing we've learned in biology, and I've tried to hammer into y'all, is the fact that things have to stay in balance, don't they? You have to stay in balance. I mean, that doesn't mean they don't get out of balance. It doesn't mean that sometimes we don't make them get out of balance. The sodium, sodium potassium pump, right? Your cells actually force an imbalance. Mother Nature tries to correct it, which we let her, and we get something out of it. We get some work done, okay? But all in all, you get hot, you sweat, right? Yeah, you get hot, you sweat. You get cold, you freeze. No, you get cold, you shiver. Anyway, you all understand. What's the word for that, by the way? What's the word for that balance we're trying to achieve? Eventually you can't, though. Oh, you fall off balance, you die. Entropy. Yeah, eventually. But what is that balance thing called? Y'all remember? It's a word back from, I think, probably certainly the first week of class. Oh, it's got such a cool word, too. Yeah, there you go. Homeostasis, right? It's a neat word. I like that word. Homeostasis, all right? Now, balance is achieved in many ways. And of course, whatever way it's achieved, no matter how you achieve this balance, we call it regulation. That's how you achieve a balance. You have to regulate things, don't you? Regardless of how you do that. So let's talk for a second about regulation. Now, I say for a second because actually, as we go out throughout the semester, I'm going to keep coming back to this idea of regulation to keep you reminded that things have to have a balance. Yes, extra credit. And you spelled it right, too. Awesome. All right, so this whole idea of regulating an enzyme, this whole idea of regulation is going to be something that we repeat over and over. Now, let's think about how we might regulate an enzyme. There's actually more than one way to do it, but let's start with what might be the easiest way first. I mentioned to you that substrates are specific for an enzyme. They fit, lock and key, okay? But I also told you that there are things that bond to enzymes, but they're not called substrates. In other words, they can fit into the active site, sorta, kinda, but the important thing is they're not turned into anything. They're not made into products, so they're not substrate by definition. Boom, not substrate. But what we could call them, though, is because they're in the middle of the active site. In other words, they're blocking the substrates, right? If there's something in this spot right here, some molecule that's stuck in there, then the substrates can't bind. We could call that an inhibitor, couldn't we, right? And there are two basic types of inhibition. One is kind of a modification of that. So let's look at that one that I'm kind of referencing. Let's look at something that binds to the active site, right? But it's not turned into product. Now, you got to remember the random nature of which these molecules come together. Right? Enzymes and substrates just happen to bump into each other. I don't need to go off on a tangent of that. We've talked about it. But enzymes and things that aren't substrates, things that are inhibitors, things that do bind. Now, some things don't bind at all. Don't worry about those. But some things that do bind, they don't stay bound forever. Enzymes and substrates bind, and then enzymes and products are released. And then enzymes are ready to go again. Enzymes and inhibitors bind. Okay? Nothing happens, no product. But then they unbind, right? So it doesn't mean an enzyme is permanently out of the loop when it's inhibited. So let's take a look at the first one. And the first one is this one, the one in the middle, actually. So there's normal binding, the way it normal. There's the substrate, fits into the active site, everything's cool, blah, blah, blah. But along comes this blue molecule. Now, does it have to fit exactly? No, all it's got to do is block, and it does. Now, don't look at C quite yet, because I got to explain something about B before we go on. I mean, you look at that, and it's simple enough to understand, right? There's something in the way. You can't bind the substrate. You can't bind the substrate. Can't make product. The competitive inhibitor, see, it's competing for the active site, right? It's whoever gets there first. Now, if the substrate gets there first, it becomes product, and then they fall off, and boom, boom, the game is open again. The contest is open again. The competitive nature is open again, and then sometimes an inhibitor binds. Now, as I said, they don't stay bound forever. It falls out eventually. But the thing is, it's not turned into product. So it doesn't have to fit exactly in the active site. Obviously, all it's got to do is block. Now, how is that regulated? Right? So that's how we stop and start the reaction. But how do we Oh, it reminds me when I was teaching my daughter how to drive. My daughter had quite a lead foot. I had to remind her that there is a big variability between stop and full speed. After a few tickets as she got older, I think she figured that out. But that was always something I said, you know, this is a variable speed car, Stephanie. You can go half the speed limit or, you know, half of over the speed limit and not get a ticket all the time. She finally figured it out. And that's what I'm talking about. Not the stop and start, but how do we regulate? How do we get that variable part in between? All right, let me do that with an example. Let's imagine we have an environment, and there we go, it works. And in that environment, I'm just going to abbreviate, we have, let's say, 100 enzymes per density, whatever, let's make it simple, all right? And in that same environment, we have, let's make it real easy, 100 substrate. Very simple enzymatic reaction. Enzyme substrate, enzyme product, boom, boom, we're all done. Now, let's add the inhibitors, and I'll say those are CI. Let's say the cell, which can move things around, right? Can move things, now it's not, goodness gracious. The cell can move things around, can import things, right? We looked at endocytosis and all those. It can even make things in its own ribosome, whatever. It could make molecules that are considered competitive inhibitors. It could make, it's supposed to be a C, it could make molecules that bind to the active site. So let's say in this particular situation, what I mean by that, this particular physiological state, for whatever reason, at this particular time, the cell is interested in slowing down the enzyme. Slowing it down, variable speed, slowing it down. So here's what it does. It produces, imports, however, it presents into the environment, say something like, let's just be absolutely ridiculous, right? Let's say 100,000 competitive inhibitors versus the 100 enzymes and the 100 substrate. Now, if you were a betting person, that's supposed to be a comma, if you were a betting person, would you bet that the next thing that's going to happen is a substrate binds or an inhibitor binds? You're going to bet. I mean, of course, there is a possibility of one, but there's certainly a much greater probability of one versus the other, isn't there? Since it's all random anyway. Oh man, I was doing good there. Couldn't have lulled you guys into sleep. So you reach into a jar, there's 100 blue, there's 100 red, but there's 100,000 green. And someone says, bet which color marble you're going to pull out. What are you going to say? Am I still online? Don't think that's a color. Are you trying to say inhibitor? Okay. Absolutely. Now, if I reverse that, if the cell then does something where, for example, it increases the enzymes or increases the substrate, whatever, if it changes that number, can you see how that would affect the ability of the enzymatic reaction to increase? So if we increase the number of substrates, or say decrease the number of inhibitors, exported them out of the cell, sent them to meet lysosomes and destroy them, whatever the physiological needs, you see how we could turn that back on again. So, as you go in through nursing into A&P, you'll see a lot of that going on, right? You'll see an awful lot of that that goes on in cells where there's this balance that's kept. And the way reactions are controlled, regulated, is by varying that balance, by adding molecules and subtracting molecules and kind of just, that's what I'm talking about, that delicate balance. So I'm talking about that really delicate balance that living systems do. That's what just always has blown me away. So any questions on competitive inhibition? It's easy enough to understand how it works. It's easy enough to understand how it's regulated, right? It's all about numbers. Let me erase that. All right. Good. Now look at the next one over there. Next one's is simple to understand. It just happens a little different way. You notice that this particular enzyme has two little notches. The one at the top that we've been looking at, which is the active site, and then some enzymes have another notch down below. And notice that it has a shape. Again, it's probably not, well, it's not square-like. But anyway, the point of that is there's something called, at this time it's not a competitive inhibitor because it doesn't compete for the active site. What it does is it binds to the other site. Now, this other site is known as the allosteric site. Allosteric. I don't think my chat box records, so I try to remember to. My pen doesn't work, and I use my mouse on my right hand, but I'm not right-handed, so it looks like garbage. Allosteric. That's the allosteric site. Some folks call it the non-competitive site, whatever. Sometimes it's even described. Sometimes people say at a site away from the active site. All right, so this non-competitor, which can be varied in number, all right, can bind to the allosteric site. Here's how it inhibits the reaction, though. It doesn't get in the way of the substrate, but it causes, you can see in the picture, it causes what we call a conformation change. Proteins are shapes. So if whatever that non-competitive inhibitor is binding to that site, it can cause that protein to flip a little bit. And in this particular case, what, that little lip is sticking out further now? Anyway, whatever happens, the substrate doesn't bind. So it changes the shape of the active site, the non-competitive inhibitor. When it falls off, guess what happens? What do you think when the non-competitive, which it will, when the non-competitive inhibitor unbinds or falls off, what's going to happen to the active site? Ryan? Sorry about that. Ryan, how about you? Perhaps Ryan went back to sleep. All right, David, then what I was asking is when the non-competitive inhibitor unbinds, all right, when this thing unbinds, when that unbinds, what do you think is going to happen to the active site? Remember, it's all about shape. That substrate's got to bind, and right now it can't because this little thing's sticking out like that. But when it doesn't have the non-competitive inhibitor bound to it, look at that. See, now if you could erase the competitive inhibitor, notice the shape, though. It's the proper shape. As soon as that non-competitive inhibitor unbinds, that little lip thing is going to go back into shape, and now the substrate's going to be able to bind. So it does the same thing, right? It still inhibits the reaction, but it doesn't do it by getting in the way like the competitive inhibitor does. This is just another way. Like I said, some enzymes are regulated this way. Some enzymes are only regulated this way. Some enzymes are regulated both ways, through competitive and non-competitive. So it's a switch, if you think about it. It's an ability by changing the amount of molecules, the number of molecules, to get this reaction to speed up and slow down. If I wanted to slow down a non-competitive inhibited reaction, if I wanted to slow down that enzyme in that last picture, I would add a lot of non-competitive inhibitors to it. And then if I wanted to speed it up, I would take them away, or I would add more enzymes or more substrates, which I can do that. So there's all these different ways in which this whole thing can be balanced out. So if you think about it, it's like one of the real fancy stereos back when I was a kid where there was a million buttons that controlled frequencies. I remember a friend of mine told me who was into stereo said that they control frequencies you can't even hear. I'm like, then what's the sense? But there's all these little buttons and knobs along these molecules that you can tweak with, because you're trying to keep, and that balance is very delicate. Can't let it get too far out of balance sometimes. Okay, does that make sense? We good there? Ryan, are we good there? Got it? Cool. Don't, it's like sometimes folks tend to make things overcomplicated. When it comes to enzymes and their substrates, it's literally just about like puzzles. It's like puzzle pieces. It's just like they're dynamic puzzle pieces. You can change them. You can move them around. Okay, so we call them three types of inhibition. So we've seen two here. We've seen competitive. We've seen non-competitive, which is also called allosteric. And remember I said it's also sometimes described. So if it says the inhibitor binds at a place away from the active site, that's an allosteric inhibitor by definition, if you look at this picture. If you look at all of this so far that we've gone through, it's almost tempting to think that it's simple as enzyme, substrate, enzyme, product, we're all done, but it's not. And if you think about the way the real world is put together, it makes sense that most enzymatic reactions are basically just a cog in the wheel, right? Like if you build a car, you don't build it all at once. So oftentimes what happens is you got a series of enzymes, in this case, four, and we're going through a series of steps we're trying to make that end product. So it's kind of like in a tooling factory. You know, they first they shape it roughly, then they smooth it off, and then they continue, and then they continue, you know, to the point where they finally get the final end product. So they're adding a little bit each time. Maybe one enzyme, maybe the reaction is so complicated, you know, one enzyme can't handle all that. Here, hold on one second, folks. I'll be all right. Sorry about that. Barking dogs. Okay, Ryan. Yeah, the recording's on, so you should be able to see that. All right, so like I was saying, it's if you think about it, most of the times, it makes sense that it's going to be something like this, right? I mean, some of the molecules that are being produced in the end product are pretty complicated, so you can't make something like that all at once. All right, so the reason I bring this up is, well, twofold. First of all, to show you that most enzymatic reactions are this way, and the other way is to kind of hint as to a way you could inhibit this. You see, involved, and this is again something you'll learn about a lot in A&P. It's called positive and negative feedback loops. But in living systems, there's nothing there that has the ability to think. And what I mean by that, there's nothing there to flip a switch on and off. So sometimes the way complex reactions among enzymes are regulated is by quantity. And in this particular case, the best way, or in most particular cases, if you've seen this example, the best way to regulate an enzyme is to regulate it by the product it makes. Think about what I'm saying. So in this particular set of reactions, the way you could regulate, and the best place to start would be at the beginning. Why start halfway through? Why build half the car and then stop? Why not regulate enzyme number one with the quantity of the end product? Now, these seem kind of simple, or not simple, it seems silly to do that. It seems silly to go through 4 steps to make the end product, which is going to shut down the first enzyme. But the important thing is to remember, whatever that end product is, that there is something called a sense of chemical inertia. Now, we're familiar with inertia. In class, I would push a chair across the room, and then with wheels, and then I would stop. The chair would keep rolling. There is a chemical inertia. So if we signal to enzyme number one, let's just kind of say signal right now. We'll talk about how we do that in a second. Stop. Stop making end product. All right? Enzyme number one would shut down. But guess what? There's still intermediate substrate A, and we didn't tell enzyme number 2 to shut down. Intermediate substrate B, intermediate substrate C. So we still have stuff in the pipeline. You understand what I'm saying? So think about that stuff in the pipeline as being excess. There's no more getting into the pipeline, but that is the stuff that would dribble out of the hose. Oh, that's a good analogy, Steve. After you turned it off. So that's kind of what I mean by inertia. Does that make sense? So think about this excess end product. And that's the important thing to talk about. All right, so this is the last kind of type of inhibition. Now, it's no different in terms of what binds to what, but it's different in kind of how it works if you think about this as being more than one step. So here's a great example. Up at the top is something called threonine. Threonine is an amino acid. Certain amino acids in your body you actually have to make. You can't assimilate them. You can't take them. You eat them perhaps, but your body doesn't assimilate them. So what it does is when it needs them, it turns one into the other. All right, so in this particular case, this series of 5 enzymes is going to turn threonine into isoleucine. That's the end product. So that's that purple, hexagon-shaped thing was in the previous picture, right? And there's a bunch of enzymes in between. So taking the previous picture, and we just simplified it. couple of things I want to point out, though, before we go on. Look at the name of that first enzyme. Remember I told you the names of enzymes give you hints as to what they are, what they do. Notice it does end in ASE. What do you think threonine deaminase does to threonine? If you were to just take a educated guess, what does that enzyme do to threonine deaminase? Yeah, there you go. That's exactly what it does. See, there's a couple of things you can get. Not all enzymes are as nice as that. Some of them have really bizarre names, but the few of them have names you look at, okay, I see what, and then when you look at the reaction, you go, okay, it makes sense. Now, I don't expect you to think about the reaction now, but who knows? Maybe you'll have a biochem kind of thing you have to look at. Regardless, it takes, 5 enzymes to turn threonine deaminase into isoleucine. Now, what about those intermediates when we turn off the first enzyme? You can see it's being inhibited. That's what they're trying to show you over here. It's being inhibited. But let's go back to the excess. Let's say the cell puts out an order. It says, all right, cell, machinery, I need 1,000 isoleucine. But there is, some in the pipeline. Let's say that there's, 200 of them in the pipeline. So when the cell finally reaches that point of 1,000, when the cell finally takes delivery, if you will, I'm kind of using a business thing, of the 1,000, there's 200 excess. The cell only wanted 1,000. The cell's only going to use 1,000. It doesn't need 1,200. It only needs 1,000. The other 200 were the ones that were they're the excess water that dribbled out of the hose. Now, that isoleucine just doesn't sit around and wait to be used again. What it does is it says, oh, this must mean, that's the thing that's flipping the switch, so this is not a mental thing, but this excess, the cell reads, must mean that I have enough isoleucine. So now I'm going over the imbalance, and I'm getting ready to put it back into balance again. So sometimes the way balance is maintained is by overshooting and under. So think about balancing in living systems as being a dynamic sort of average thing. The excess, as you see in the picture, and I highlighted in red, go back allosterically in this case, and many times that's the way it is, and change the enzyme, right? And they change the enzymes through non-competitive or allosteric inhibition. Regardless of how they do it, what they do is they inhibit threonine deaminase from doing anything, because see the active site at the top, right? The active site at the top, if that active site has been changed, then that no longer can do anything. It can no longer bind to threonine anymore when it looks like this. Oh my, now my pen's working. And then how you turn that back on again, if you're going with the full circle of regulation, is you see the arrow going to the left, then the isoleucine says back to the machinery, hey, you remember that 1,000 isoleucine that you made for me a while back? Machinery says, yeah. It says, I need another 1,000. Oh wait, here's 200. Boom, I take those. So it immediately removes all of those things that have been acting as inhibitors, and the cell starts cranking out another 1,000. So you always get this excess, right? And that excess can be regulated. So, see, that's, if you think about the way, this is what we would call positive inhibition. You're going to learn about that in A and P, because that means that excess of something is causing something to occur, an inhibition to occur. Isn't that kind of neat? So there's never any magic to any of this. It's all just kind of makes sense. Like we were talking about inhibition in general. It's all just about numbers. I think this is ingenious for a couple of things. What's the best way to regulate something? Why make an additional molecule called an inhibitor in this particular case? Why not make the excess, why not make it not a particular molecule, but the amount of molecule, the excess is being the inhibitor? Find that brilliant. Okay, no questions on that? Then the next 3 things I want to talk about are nice because they're going to fit in with the lab. We're doing lab 6 next week, by the way. I put an announcement up. We're not doing 7. Can we just do 5? Yeah, I think that's what I said in the announcement. Same thing. Absolutely. Keeps the flood, keeps the tub from overflowing. Keeps it regular. Keeps that water level, assuming that the water coming in isn't faster than the water going out. But yeah, exactly. No, not this Monday. Is it this Monday? I want to wait for 5. Let me take a look. I'm horrible with dates, so I'm always in the habit of looking at someone. And this is the 4th. Yes, then there is an exam, chapters 4 through 6. So we're finishing up chapter 6 today. Lab 6, that's the one we're doing, not 7. Yep, so your assignments are due 4 through 6, and we're finishing up 6 today. So those are due, but that'll be on the exam Monday. I know that's a little bit tight. Yeah, I know it's a little tight, but that the hurricane. After that, we'll be in good shape, though. We shouldn't have any issues as far as time. Thanks for reminding me of that, so I put that in recording. Okay, so the lab that we're going to be doing is going to, you already know the answers to these, honestly. The lab we're going to be doing is going to be looking at what we call the effect of local conditions on enzymes. In other words, here's a way that enzymes are inhibited, but here's another way. So it's another one of them switches, right? There are ways that we affect how fast an enzymatic reaction works. That's what we're talking about. Enzymatic reactions are kind of like your gas of your car, or like the engine of your car. If you add more gas, the engine goes faster. Nothing, if I said that and that was the end of today's, as far as enzyme, it makes perfect sense, right? Add more gas, car goes faster. That's what my daughter was figuring out. So, can your car go infinitely fast? I mean, if I keep mashing on the gas pedal, will I keep going until, well, at least Einstein said the speed of light. Something I read the other day said there is things that actually go faster than that. But no, you can't go infinitely fast. Could you run home infinitely fast? Could you run anywhere infinitely fast? Ryan, you could probably run pretty quick, but can you run infinitely fast? Well, you'd be in the NFL if you could, right? I mean, you have an upper speed. It's one of those things. Yeah, not enough energy. Legs aren't long enough. I like to run. I'm a long distance runner. I'm never going to win those races. I'm a short guy. The guys who win are the guys with the huge long legs, or the guys who are in a hell of a lot better shape than I'm in. It becomes physics in that situation like that. So if you think about that, enzymes are the same way. They're cranking out stuff. Yeah, there are. That's right. I read about that. I didn't quite understand how they proved it. Yeah, but those are the things that are trying to collect underground, aren't they? The things that that's really, really heady. That's the stuff that I look at and I only understand a little bit of it. So substrate concentration down below, whatever. Substrate is gasoline for an enzyme. On the y-axis is the dependent variable, isn't it? Isn't that the dependent? Look, if you word this, remember I said sometimes if you word it, particles, like through, what is that called, intertwining, or the fact that particles are intertwined across space. Is that what you mean? Yeah. That's what I mean about quantum mechanics, the fact that we're all in the same place all at the same time. Does this make sense the way it's plotted too while we're talking about that? Does this make sense as the y-axis being the dependent? Say it to yourself. The reaction rate depends on the substrate concentration, or does the substrate concentration depend on the reaction rate? The important word is depend. See, that's what I mean when you're trying to figure out independent and dependent. When you word them, many times it just makes sense. Is the speed of the car dependent upon how hard I mash on the gas pedal, or is how hard I mash on the gas pedal dependent on the speed of the car? One makes sense. So look at this particular enzyme. It's turning substrate into product. That's what the reaction rate is. But you notice that about 1, whatever that concentration, you get a leveling off. Can enzymes turn substrate into product infinitely fast? No, of course not. They reach a saturation point. That's what that is. They reach a point, you add more and more and more, and they go faster and faster and faster. Substrate, they eventually get to a point where they're in the weeds. They can't go any faster. There's nothing bizarre about that, is there? Now, the next two ways or next two local conditions that can affect an enzymatic activity, you have already seen the idea behind it. You already understand the why. So I'm going to show you the graph, and then you guys are going to help me understand the why. But does everybody understand what I mean by this? In this particular case, the local condition that's affecting the enzyme is the amount of substrate. It can only go so fast. Things are not infinitely fast. Right? Sometimes I look at this stuff and I go, guys, this just makes perfect common sense. Just, stop making it more difficult than it is. Is that what I said? 6? Yeah, I believe it is. It's on. I just posted it to Blackboard. Yeah, 6. No, go on Blackboard and look what it says 6 now. Are you on Blackboard looking at, I didn't post the wrong one. Just reading it this morning. Lab 6 should be titled Enzyme Activity Testing the Activity of Catalase. Okay, good. Ooh, you got me freaked out there for a second. I've been known to do things like that. No problem. All right, this makes sense, doesn't it? Can only go so fast. All right, look at this one right here. This is the one. This is temperature and reaction rate. Now, I will give you a little sort of lead into it. We talked about molecules bouncing off each other randomly. One of the ways you can get molecules to increase the probability of running into each other is have them bounce off each other faster. And one of the ways you make molecules go faster is by putting in energy, like heat. So that's why you see that gradual rise, up, 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 0, 5, 20, 25. The rate of reaction is going up. The more it warms up, it's like photosynthesis is this way. When leaves heat up, they photosynthesize faster. What I want you to explain is what happens after, about 37 degrees Celsius. Why does the reaction rate drop? It does, but why? This is what I always warn students about when they're explaining graphs to me. If I said, what does the drop in the graph tell you about the enzymatic reaction rate and all that? And they say, well, then it went down. No, I know that. I want you to tell me the why. Why do you think at that really warm, 37 degrees Celsius is, okay, faster temperatures, particles move real fast, but 60 degrees, there ain't nothing going on. 37 degrees is about body temp. It is body temp. Why do we have the peak and the drop? What's happened to the enzyme? What is an enzyme? What are those things that could affect an enzyme? Well, they might, but even before that, what's happening to them? What's happening to them is you're getting them warmer and warmer and warmer. What is that word that we introduced back in chapter 3? Condition that happens to a protein, which of course their shape is, absolutely crucial. Yeah, they're being denatured. Do you remember back then there were a couple of things I mentioned in class, and I said that you're very familiar with denaturized proteins. I said these are two conditions that'll do that. There was eggs, and how did you denature egg protein? What did you use? So what is happening there? That's exactly right. What's happening there past 37, 38 degrees of Celsius? What's happening to the enzyme? It's getting pretty warm. It's denaturing, and if it's denaturing, that means by definition it's changing its shape, right? Or its shape's being changed. And if its shape's being changed, so is the active site. And so now, if the active site shape is being changed, what's happening to the substrate? Or what's not happening to the substrate? Sure, can't fit in, right? Simple as that. So that's what I'm talking about, kind of remembering back. Again, I don't give comprehensive type exams, but all this is so very comprehensive. I might ask you on an exam or on a lab practical exactly what I just said. Tell me why the reaction rate increases and then drops off. Or I might say something like, at what temperature is this enzyme starting to be denatured? What would you say to that? Well, you could look anywhere along that curve. But you would say somewhere around 35 to 40 degrees, we're starting to denature it. Now, that enzyme is almost fully denatured, it looks like, if it's not fully denatured at 60 degrees, isn't it? At that point, what David said earlier, it probably does start to break apart, start to destroy it completely. Before that, you're getting it to twist and change, and of course that enzyme is resisting that. But remember, heat is energy. Remember what it did to water molecules? Bling, kabling. Oh, my springs are at school. Kabling, kabling, kabling. So the same thing's happening here. That molecule is binding and twisting it, and then eventually it's just changing out of shape. So we had substrate concentration, we had temperature. Here's the last environmental condition. I said there were 3. Substrate concentration, it overwhelms it, can't go so fast. Here, the explanation of the results is the denaturing of the protein. You explain this one to me. It's a little bit different, although it is similar to what we just saw. What's happening below 4.5 and above 4.5, where that arrow is? So 4.5, whatever, some number there. What's happening? Don't tell me the reaction rate is dropping. I can see that. Why? What's causing that to happen? Yeah, it's denaturing it, isn't it? You're not getting reaction. You're not getting now. Okay, you might have been saying, I don't know what's happening. Maybe the protein is just exploding. There are lots of things that could be happening. Okay, one of the things that we would suspect, though, if we saw this, if we took an enzyme and subjected it to different pHs like this and got a reaction rate that looked like this, we might suspect, hypothesize, we might deduce, we might ask a question: is it denaturing that's causing this to occur? That's very reasonable. Yeah, you both got put it together. It's becoming too acidic or too basic for the enzyme. Remember I said things that denature proteins, enzymes, are heat and pH. So in this particular case, we can talk about, for example, an optimum. What is the optimum pH for this enzyme? What's the pH at which it works the best? That's what we mean by optimum. Yeah, 4 or 5, there's something like that. Let's go back to the previous one. What's the optimum temperature for this enzyme? When do you get the greatest amount of reaction just before, oops, we broke it? My son and I built a little mini trebuchet, and we were launching golf balls and all that. Of course, then we decided to see how much we could launch, and then we broke it. So we overwhelmed the trebuchet. Yeah, somewhere probably around in there. Yeah, that'd be a pretty decent guess. It looks like it starts to level off, and then it begins to denature it. Sure. Now, is that the only reason why it's doing it? No, there are those other reasons, but that's see, this is science. Then we would do additional tests to see if they're denatured. Now, look at this. This is good evidence for this. At the top is what, temp? Yeah, at the top, we have the temperature, and we have an optimal curve. So again, this is like for the one at the top, it's temperature. You see the curve kind of rises because it's getting warmer, molecules are moving faster, and then, oops, we broke it. That's the little blue arrows that are about there. That would be the optimum for those. What I want you to notice about this is kind of two things. Being able to read the optimum, well, that's simple enough. Just that's at the top of the peak. So we got 37 for the one in the black. And what, 77 for the one on the right. What I want you to notice that's really cool about this is the fact that where these enzymes are found. The one that's in the black is an optimal temperature for your typical human enzyme, and the one in the red is the optimal temperature for a bacteria that loves heat. Thermophilic means to love heat. They live in those, oh my gosh, they live in those geothermal vents out in Wyoming and down around those ones at the bottom of the ocean. Some of them live at temperatures above 100 degrees Celsius, which is boiling. Notice how the enzyme's optimum rates. You would think, oh my goodness, the enzymes in the bacteria at 77 degrees must be denatured. And Mother Nature's already figured that out. She makes the optimum temperature for an enzyme about where the temperature that enzyme is going to normally experience. Now, can you denature that typical human enzyme? Yeah, when people get too warm, heat stroke, that's what's going on. Enzymes are denaturing. It's one of the things that's going on. Same thing with the bacteria. You could slow it down and you could denature it. Oh, yeah, those tube worms. Tube worms have no gut. They don't feed. They actually live with bacteria, these thermophilic. Not all smokers are hot. There's some actually cool, but the really black, the black hot smokers are close to 500 degrees Celsius. Weird. Look down below, same thing for pH, right? So if I said the average pH of the human stomach is 2, what do you think the optimum pH for an enzyme found in the human stomach would be? I know it sounds like a trick question, but it's not. It's a Z. Yeah. How about for trypsin? Trypsin is another one, but it's found in your intestines. So the optimum. So what do you think your average intestinal pH is without using a pH probe? If I said, all right, down in the intestinal region where trypsin is found, what do you think that, yeah, and the same thing for the stomach enzyme, right? I trying to show you there? I'm trying to show you how Mother Nature designs all this randomly, but all this is then selected for to fit perfectly in its environment. This, at the very molecular biology level, is an idea of how things adapt to their environment. Now, enzymes aren't alive. They're not adapting, but the organisms that contain them are. And so it would make absolutely ridiculous sense for an enzyme that's found in a human to denature at 20 degrees, or to not only be active until you reach 60 degrees, right? It all fits together, form and function. All right, did y'all take your vitamins today? Oh, it's like children or adults don't need vitamins. I'm like, yes, they do. Unless you eat well. And I know there's some talk about whether they're good for you or not. I've always taken vitamins all my life. I don't know. Is that right? Do we always get the right amount of even some of those trace elements? Because I'm not a very good eater. I never have been. I mean, I eat good food. Well, that's some case. I know in vitamin C, but I don't know if that's in all vitamins. Yeah, I think there's probably a. I've heard about that. Well, human beings being what they are, I do know there's that tendency to overuse things. Oh, gosh, 10,000. That's why sometimes I look at the vitamins and I go, do I need 10,000 units of this? It seems like it's 333% above. I'm assuming those are the ones that are water soluble. Yeah, I kind of go back and forth on that. I've always taken them, but I don't know if the fact that I've been able to stay relatively healthy has as much to do with genetics as anything else. Interesting. Scientific community changes. And I think that's just as our knowledge grows. Yep. I think it does too. My grandmother lived to be 99 and she wasn't anything. She was not in shape. She loved fried food. Her doctor, she was 90-something. 93, and he said, her name was May Jones. He said, you're going to have to stop eating fried food. Why? It's bad for your heart. She goes, I'm 93, doctor. Speaking of vitamins, all that, there are things called cofactors or coenzymes. Your body does naturally produce vitamins too, so David's absolutely right on that, of course. But when we talk about things that are cofactors, you actually need a little bit of some metals like zinc and magnesium, which I've included in the picture down below. You need these would be trace elements. Like, for example, if you remember back in hemoglobin, there was an element in there that you needed, right? Iron. That's why women are potentially prone to anemia. Anemia is the lack of your hemoglobin to carry oxygen, and the reason sometimes why that happens is because of a low iron level. In order to form that hemoglobin protein, that big quaternary structure, you got to have iron. So women are low on iron, and then they lose blood, obviously, on a 28-day cycle. They can be careful if they're not about anemia. I believe that. Yeah, trace element. This is the one that always freaks me. I think it's selenium. If I remember right, selenium is the one where you need like, some millionth of a microliter or some weird number like that. But if you have just a little bit more than that, you die. Yeah, I think it's one of those weird ones where you need just right in the middle. I guess it's strange. Plants, for example, they would use they would be important for them to use magnesium. Yes, magnesium in their chlorophyll molecule is the one, like our iron and our hemoglobin, that binds it together and allows it to absorb sunlight. Coenzymes, then, the difference between cofactors and coenzymes is whether they're organic or not. So the ones that are cofactors are metals. Those are inorganic. Remember, organic is it's going to have carbon and hydrogen. And those are going to be things like the vitamins B6 and B12 and all that sort of stuff. What do we use the B vitamins for? That's the energy one for the most part, right? Isn't that the thing they pack in 5-hour or 4-hour energy drink? Is they just fill it full of B vitamins and tell you it's all natural? Still tastes awful anyway. Ryan, do you have any questions for me? That's actually the last slide, but I've got time to lecture over. Then of course I'm in office hours. But if you guys you don't have any questions about the lecture, I'll stop the recording. I can stay in office hours, but I'll certainly stop the recording. That sound good to everybody? So that's the end of chapter 6. So that's what the exam's over next week, 4 through 6. Relatively short, relatively simple stuff. Good chance to bring up your grade, too. All right, I'll stop the recording.